Before we begin the real stuff, I'd like to emphasize, slightly self-centeredly actually, how much I love the COB and how much the COB has influenced my own career. I went to my first ever BSDB meeting in 1976, 50 years ago next year. And if that doesn't make you feel old, nothing will. And I spoke at my first BSDB meeting in 1978. And I've been to many meetings, of course, since then. I think I published more papers in GEME or development than in any other journal. I was on the COB board from 1986 to 2002, and I was editor-in-chief of development from 2003 to 2009. Throughout that entire time, and to this day, the COB has remained a force in biology because it is a company for biologists, run by biologists, and long may this continue. One other thing on the COB, I'd just like to add that the COB timeline that you can see downstairs mentions many of the people who've made the COB such a success. But one more name I'd like to mention is that of Simon Madrill, who was COB's investment manager from, effectively 1965 until his death in 2020. Simon increased COB's value by compound growth 18% year on year. Wouldn't we all like to have him at home? And we owe him a lot. So, Simon Madrill, not all of you will have heard of him, but he was one of COB's greats. Okay, now, to business. You will all have heard of Sidney Brenner. I think he is a genius, or was a genius, I don't think that's too strong a word, who changed every field he entered, from discovering the triplet code, nature of the genetic code, solving the code, establishing C. elegans as a model organism, introducing fugu as a model for the genome, and establishing several outstanding centers of biological research around the world. But Sidney was also a brilliant raconteur and a wonderful coiner of aphorisms. Now, everybody impersonates Sidney, I'm not going to. But let me remind you of a few of the things he said. One of my favorites, actually, have you tried neuroxing papers? It's a very easy and cheap process. You hold the page in front of you and let it go from there into the brain. It's much better than Xeroxing, he would say. He also said the modern computer hovers between the obsolescent and the non-existent, which I think, true. He said, I think one of the things about creativity is not to be afraid of saying the wrong thing. And, in this world, we're all too afraid of saying the wrong thing, and I completely agree with him on that. Say what you think, and don't worry if it might be right or wrong, because we need to hear what you think. But of them all, my favorite remark of his, or the one anyway most relevant to what we're going to talk about today, is progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. And as we reach the end of this meeting discussing the last 100 years and looking forward to the next, I think it's worth asking whether Sidney was right about the importance of technology, and that we ask more generally about how science should progress. What will science look like in 2125? What will matter to us? Will we be looking for different kinds of answers to different kinds of questions? What will be the effect of artificial intelligence and machine learning? Will humans have their digital twins? How will we improve the lot of humankind? And probably most important of all, how will we make sure that we're all here to celebrate, well, not all of us, obviously, because I'll be dead, but how will we make sure that we're here to celebrate the 200th anniversary of the COB? So those are the sorts of questions we want to think about. And to help us think about it, we've got four brilliant researchers who will bring different perspectives to the question. First, we'll hear from Nick Hopwood, a developmental biologist turned historian of science, who will begin by bringing a view of the past into the discussion, including the changing role of technology in scientific advances. So Nick will give us some history. Next, well, we heard from Jennifer Lippincott-Schwartz this morning, and she almost began the conversation that we are having this afternoon. She's an innovator, a cell biologist. She developed the use of GFP to follow cellular tracking in living cells. She used FRAP to show that molecules diffuse freely within cells. She developed photoactivatable versions of GFP, and she worked with Eric Betzig to develop super-resolution imaging methods. So after Nick, we'll hear from Jennifer. Then Maria Leptin is a developmental biologist who's made fundamental advances in Drosophila development, especially the cell movements that occurred during gastrulation, and who can bring here also, however, her extensive experience in research funding and administration through her roles at EMBO and the European Research Council. And finally, to bring a more relevant and contemporary perspective than the one brought by a group of people whose names I've just listed, whose average age is 68.5, I'd like to welcome BSDP representative Tamina Lebek and Levy, who, as I think 6 months, brings the age really down, I'm pleased to say. She's using mouse ES cells to model aspects of development, and she's developed a new fluorescent neighbor labeling system to study cell behavior. I think, Tamina, you may also want to talk about questions like career structure and sustainability, but that's entirely up to you. So each person's going to speak for no more than 10 minutes, frequently less, because the object of this exercise is to hear from you, the audience. Do you agree with the panel? What have we missed out? And we need you to speak up with suggestions, ideas, refutations, arguments, scorn, and enlightenment. All of those things are welcome. So I'm going to hand over to Nick now. I'm going to sit where Nick was sitting, and after Nick it will be Jennifer, Maria, and Tamina. Nick.
Thanks very much indeed, Jim. And thank you to the Company of Biologists for making this possible. People used to ask Brenner for references for his quotations. He traced ours to a Nature report of 1980. And then he found the notes for his talk. And you see, as students of quotations would expect, that things get snappier as they're repeated. But also that Brenner didn't just rank techniques, discoveries, and new ideas. He stressed the interplay. Brenner was speaking at the 10th birthday party of an institute which had asked scientists to look forward 10 years. The Nature report began, "Good scientists don't speculate a decade ahead of their research." By taking on the next 100 years, my fellow panelists have a 10 times harder job than Brenner. My job, looking back on what already happened, might seem 100 times easier, but it's tricky to identify drivers of change. Brenner's fellow molecular biologist, François Jacob, who also moved into developmental biology, discussed this at length. Jacob aimed, in this book, to discover how objects become accessible to investigation, thus permitting new fields of science to be developed. Jacob had helped turn bacteria into genetic systems. He was moving into mouse teratocarcinoma cells. He wanted to know how biologists had opened up successive levels of analysis, the visible structure, cells, and then in the 20th century, genes and molecules, culminating in his own idea, the genetic program. And that's certainly one big story for the century of the gene. Now, for Jacob, progress involved the interplay between observations and techniques, practices and interpretations. But for him, scientific theories contribute more than anything else to reorganizing the domain of the possible, modifying ways of looking at things, bringing to light new relationships or objects. Now, I'm a materialist. I'm generally happy to put techniques before ideas, but the balance is going to depend on the case. What about discoveries? Discoveries are where techniques and ideas come together, sometimes unexpectedly. So far, so good, perhaps, but both categories, techniques and ideas, are too narrow. We've got to expand them if we're going to consider how science will and should progress. So let me try to expand thinking about technologies first, because technologies depend on and sustain infrastructures in medicine, agriculture, and industry, and these provide access to organisms and other resources. Think of collecting through expeditions or in hospitals. Think about how cell and molecular biology really came out of cancer research, how stem cells came from fertility and abortion clinics. And of course, biology has its own infrastructures for animal supply, scientific instruments, and so on. In the wake of the Human Genome Project, these have rather tended to take over from question-driven research. Now, all of this is expensive, so we have to talk about power. Biology has moved in the last 100 years from the margins to centre stage. In 1925, this process was just beginning with, say, the National Institute for Medical Research in London. In 2025, the UK government pins its hopes for growth to a significant extent on the Cambridge Biomedical Campus. So we're seeing the £1 billion AstraZeneca building between the Laboratory of Molecular Biology and the Royal Papworth Hospital. Now, biology was already involved in state projects in agriculture and medicine a century ago. Eugenics, as you know, fed into Nazi biology and cast a long shadow. But after World War II, in what was the American century, government funding produced biomedical and breeding enterprises orders of magnitude larger than anything that had ever been seen before. Biology kept people alive, it produced food, it shaped the environment. But since the 1970s, science has in many ways been in crisis. Perhaps first, crises of being on the side of the powerful, increasingly crises also when those in power declare war on scientists who state inconvenient truths, including about our burning planet, biodiversity loss, and the health effects of pollution. Now, biology is obviously in danger of being crushed by Trump and co., but it also risks being co-opted into projects that discriminate and distract. Negotiating this, and I don't think we should pretend that this is just an academic exercise. This is going to be crucial for the very survival of biology and for the terms of that survival. What audiences can biology address? What alliances can it build? Okay, let me say just a little about ideas. Another reason to put techniques first is that technology often inspires ideas. So think of the genetic program as coming out of computers. Think of cellular organization modeled on industrial cities. Now, I could talk more about this, about ideas larger than concepts, whole structures of thought. We could say that what matters is not the answers, but the questions, or we could ask how visual images shape knowledge. But I want to close with something different, a word about identities. Biology was still barely a thing 100 years ago. The founding of the Society for Experimental Biology and of the Company of Biologists, those helped make the last century a century of biology, and biologist something that you could become. But what, if anything, will it mean to be a biologist in 100 years' time? We'd expect a variety of answers because even at this meeting, biologists come in various kinds. and there's been a huge change in who can be one, though still not enough. So we can imagine new disciplinary identities, and some of those might be just fine, but how to think about the future? Roughly 100 years ago, the biologist J.B.S. Haldane gave a lecture on science and the future. He speculated about ectogenesis, babies in bottles, curing disease, and supplying unlimited food. That was going to involve algae escaping into the oceans, turning them purple, and meaning that everyone could eat as much fish as they wanted. Haldane's lecture inspired his friend Aldous Huxley's novel Brave New World. So let me close by quoting Haldane. "I have tried," he wrote, "through these visions of the power of reason to show why I believe that the biologist is the most romantic figure on earth." Now, at first sight, he seems to be just a poor, little, scrubby, underpaid man groping blindly amid the mazes of the ultra-microscopic, engaging in bitter and lifelong quarrels over the nephridia of flatworms. But I suspect, said Haldane, that more biologists dream than would care to confess it. And what he's bothered about is, were the dreams the right size? We are at present almost, so this is in 1923, almost completely ignorant of biology, a fact which often escapes the notice of biologists and renders them too presumptuous in their estimates of the present position of their science, too modest in their claims for its future. So what's the right balance between presumption and modesty today? I look forward to hearing my fellow panellists' dreams. Thank you.
Thank you, Nick. Thanks, Nick. I'm going to go back to the lectern, but I'm going to hand over to you, Jennifer, who will speak from where you are. That's right. Great, thanks.
So, should I start right now?
Please do.
Okay. First, Nick, that was incredible. Very, very profound, exciting. What I have to say is going to be a little bit at a lower level, but very, very cool. I hope we have a lot of discussion about what Nick talked about. So, and I should say that we didn't know what you were going to be talking about. So it's so cool. Anyway, in terms of going to this quote, that's where I'm going to start. Technology, new discoveries, new ideas. Where does it start? Who's top, who's bottom? I guess my view is that no question technology is driving science in many, many ways, but science has to be ready for that technology. And I've experienced this many times in my own career, where if the field is not primed for the technology, it goes nowhere. That technology goes nowhere. And one example is FRAP. So when I was a graduate student, there was photobleaching. Biophysicists were playing around with photobleaching with crummy dyes, looking at the surface of cells, and nobody could care less. I mean, it was incredible. I remember being taught about it as a graduate student and thinking, wow, this is pretty cool that these dye molecules are moving on the surface of a cell. But beyond that, because we did not have GFP, we did not have ways to look at proteins, the field didn't think about diffusion or motion in the cell. And it was only, when we had the ability to tag specific proteins with a fluorescent marker that we could see these proteins potentially, if you look under a microscope, you see a steady state distribution. But it was only when we started employing FRAP in that context where you could actually see molecules recovering into a large bleach box. And you knew what these molecules were because they were tagged specifically, that people began to get extremely interested in terms of what that approach could bring. And that was like 10 or 15 years after the original discovery that you can use light to bleach out a fluorescent dye. So unless a field is primed for a particular technology, the technology is not going to go anywhere. Now, so because of that, I really think that science is moving through a synergy, which is what Brenner really said. It's an interplay between new discoveries, technology, and new ideas. These three different topics, if you will, really feed back and fuel each other in very dynamic ways. And so the technology can lead to a discovery, which in turn can generate new ideas that in turn lead to even further technologies. And basically, we see this all the time. I can just give you some examples. Well, I should also say that the point about that is that technologies often are not invented or developed in a vacuum. They're prompted frequently by a need in the field. And so just as an example, photoactivatable GFP, which we were involved in developing, it's not that we were just thinking, oh, wouldn't it be cool to have a fluorescent protein that could switch on? That's not what we were thinking. We were thinking, wouldn't it be really nice to be able to turn on a certain group of molecules in the cell to look at how they're moving? We have no idea how these molecules are turning over, and through GFP, we cannot easily see how they're moving unless we do some very fancy type of photo bleaching. So basically, it was the need for that photoactivatable protein that led us to actually go in and try to find it, modify the proteins. And this is how many technologies are developed. People see a need as a consequence of discoveries and new ideas, and they then create technology that will follow. So basically, I think there's an incredible synergistic interplay between these three topics that has been driving biology as well as science in general over the last 100 years. So what about the next 100 years? And that's where I think we are in for some really interesting new directions. And part of that is, in fact, I think there's a possibility of going outside this loop big time. And it's because, and that going out of the loop of technology, ideas, and discoveries is sometimes exciting, but I think it's also troubling. And that the technology that's taking us, I think, in that area is potentially AI. So AI can generate lots and lots of new information. And it can connect that information in new patterns in ways that we could not dream of. The problem is that the speed of this information gathering and the patterns and interconnections that are detected by AI can very easily outrun our capacity to draw new ideas and discoveries from it. So what that means is very quickly it's AI who's going to be generating these concepts and ideas. But a key aspect of the way that we do science is that we seek out causal mechanisms. But much of the data that AI is going to be generating is going to lack these causal mechanisms. It's only going to be showing us patterns and relationships that may have little to do with what we as biologists are fundamentally interested in. We want to explain things. We want to know underlying mechanisms, how these mechanisms are being maintained in a chemical as well as a physical world. And it's not clear to me how this AI revolution that we're experiencing right now is going to necessarily connect with the way that we've previously done science. And so that's something that I think would be very worthwhile talking about. I would love to hear what Sidney Brenner would have said on this topic, as well as Jacques Monod.
Maria.
Yes, thanks. Great to be here. I too have published in at least 3 of the COB journals, and I love them, and some of my most important work was published in them. It's funny, we're all using quotes from the past to speak about the future in a session that is about the future. But of course, the past is all we have, and its comparison with our current present is in fact what allows us to extrapolate about the future. And this quote thing is a good model for speaking about discovery and research, especially in the context of science funding. Because we know what we did in the past, 10, 20, 30, 40, 50 years ago, what we discovered then, and we know how those discoveries are now being used, either for benefit of mankind or for future discovery. But the important thing is, we didn't know that back then when the discoveries were made. We did not know what they would be used for today. Fleming, when he saw fungus growing on his bacterial cultures, didn't think, yes, I can make that cure pneumonia in the 1940s or so. And Francisco Mojica and Eugene Konin, when they discovered and then studied CRISPR sequences, didn't think, wow, a lab in Cambridge in 2025 will be using this to make chimeras between mouse and jerboa, as we heard this morning, which I thought was pretty cool. So no, they didn't. They only cared about what they were discovering then, and we only know with hindsight what that can be used for. The important context, like I said, is funding, and policymakers really don't get that. They usually want to focus, and of course they have to focus on current challenges, on current programs. They're spending taxpayers' money and they care about their voters. Even so, they don't get that what we did then is what helps us now, and therefore what we are doing now will be helping us in the distant future, maybe. A clear case of this, by the way, is AI funding, where everybody is going mad and out-competing each other how much money they want to put in, whereas the AI geeks are, saying large language models are passé. They're long into completely new ideas, completely new thoughts, and it's those that we should be funding. So back to the theme, techniques. Can we force the invention of new techniques? And Jennifer, I've written this question down. Jennifer just answered it. No, of course not. There is so much serendipity involved, not just in whether a technique that you might stumble across is going to be useful, but also in whether the technique that you need today can be found, and who you bump into and who you talk at a meeting. So that serendipity is important, and planning is really hard. Scientists invent new techniques if and when they need them. Now, I want to make one and so I'm not prepared to make any predictions about the future. I'm in favor of funding scientists, the best scientists, for their best ideas that they have now, and trust the scientists that they are the ones who know best where the frontiers are that they need to break through. But one final concern, Manu Prakash made very clear this morning that we must make available techniques and methods as broadly as possible, and it's of course admirable what he's been doing in that regard. And some new techniques and methods do become broadly usable immediately. I mean, PCR was in labs within no time flat, and similarly CRISPR. You know, when politicians are still proudly talking about projects that use CRISPR, in fact, there's nobody who doesn't use it. So some are really fast, but the concern is about the large, expensive infrastructures. We're used to it from astrophysics and from physics, CERN, or the huge telescopes, but we're seeing it now. I mean, the work you do, Jennifer, needs a Jennifer to do it, and needs a Jennifer's insight and creativity, but it also needs HHMI money. And so it tends to happen in big centers. that have the infrastructure and that have the methods, and it cannot happen in the more peripheral institutions. So it does create now in biology a new way of doing science that other fields have long been used to, and I think that's another consideration that we have to take into account when we think about the future.
Thanks, Maria. So I think that's going to raise questions, Mia, about things like what is the word? Democratization of science, I suppose, is what we're talking about. Tamina, over to you.
Right, thank you.
Thank you for coming.
I'm really grateful to be here, and not just to drive down the age average, but also that I can be here with Levi. But basically, I think we've established that developing new techniques is a bit risky, and especially when you're trying to squeeze it into a 4-year PhD, which also comes with the nature of funding for PhDs. I learned this the hard way. When I started, I very naively thought that I'd spend a year, maybe 1.5, setting up my neighbor labeling system, and then I'd have 2 good years left to use it for real discovery. Reality, of course, is always different, and I was 3 years into developing, optimizing, and troubleshooting when I had just 6 months left to actually apply the Puffin system to something before going off work to spend some time with my side project in development. Yes, you, exactly, I'm talking about you. And yet, I still consider myself very lucky because it worked. And the truth is, technique development is a high-risk, high-reward game. So sometimes I wish there was some kind of fairy godmother specialized in techniques for developmental biologists, someone who could just, wave a wand and conjure up the ideal technique. What would that look like? For me, the dream would be a system to kind of track the phone records of cells, who's been talking to whom, and ideally what they've been saying in both development and disease. What would your ideal technique look like? But beyond our individual wishes, what should we strive for when we're developing new techniques? And I've got a wish list for that. First of all, I think we need transparency. I think we need to clearly communicate. Exactly. I think we need to clearly communicate not just the strengths of our systems, but also its limitations, so that scientists can carefully evaluate if it might work for them in whatever setting they have. That leads me to the next point, which is accessibility. Sharing protocols, sharing knowledge and training resources to ensure that others can build on this work is key. The third thing I want to bring up is affordability, because this is so often a problem with new techniques. We need funding, we need money, especially when something is new. But I hope that science isn't always limited by funding inequalities, and that we can promote, or that we need to promote, resource sharing and open access wherever it's legally possible. And lastly, I want to mention sustainability, which has come up a couple of times during this meeting, and the Company of Biologists is really great in promoting this. So if we have a new technique, can we also think about how maybe that technique can reduce waste or can make our experiments actually more efficient? For example, can we use AI to streamline our experiments to find the best starting point for those? But also, coming back to collaboration, tell other people what has not worked for us so that maybe we prevent duplication of things that just won't work. And these are increasingly urgent questions. But above all, I think a technique is only as valuable as its impact in the end. If it's not useful, if it's not used and adapted, it won't drive the discoveries and ideas that we need to push science forward. Looking back, I'm incredibly grateful that I had the chance to develop a new technique early in my career. But just as important, maybe more, I had the opportunity to share it. Every time I've presented my work at BSDB meetings, and this is my fourth one with a poster, I've come home with new ideas and insights from talking about it. And our published paper is stronger because of the collaborations that we built after posting the preprint. Again and again, throughout my PhD, I was reminded that science moves forward not just through the techniques, but through the people who create, refine, and share them. So that's really my final lesson for this introductory part, that the best techniques, like the best scientists, embody what we value in good academic citizenship: authenticity and purpose, inclusivity and collaboration, and a commitment to continuous improvement. So in the end, I do think that Brenner was right. Progress in science depends on new techniques, new discoveries and ideas, probably in that order. But I'd argue that behind every technique, every discovery, and every idea, there are people willing to take risks, to share knowledge, and to work together. And that's what drives real progress. What do you think? Right, thank you.
Thank you, Tamina. So this is the bit that I think we've all been looking forward to, which is to hear from you. Now, standing here, I can't see very much unless I do this. So I'm now going to do this, and now I can see a bit more. So members of the audience, if you have something to say, we'd love to hear it. So please raise your hands or move somewhere towards a microphone. I can see some people beginning already. So I'll be right with you. Anybody else? Okay. Number 2, as I'm going to call you.
Yeah, so when I saw that this awful quote is going to be the topic of this session, I resolved immediately to make us think about it, right? And of course, the panelists have taken the wind out of my sails, right, by talking about how really it's the interplay between these things that matters and how it's the people behind these things that matter. But, you know, for me, the fact remains, this remains an awful quote because we bring it up all the time. And maybe, you know, the younger people in our sort of club, myself included, we don't have the insight of our panelists to sort of interpret this quote in the right way. And I think this you know, the quote is sort of emblematic for a culture that devalues ideas compared to techniques. And this may, in no small part, be a reason why, by my estimation, we now are in a time where, you know, we are drowning in data and starved for insight. So I would suggest that we maybe retire this quote, and maybe we can think about alternatives. Right? I wasn't going to make a submission myself, but then the topic of dreams came up, right? And I was inspired. So maybe progress in science depends above all on the dreams of scientists, and especially on the freedom of scientists, on the freedom of different scientists to dream different dreams. Thank you.
Thank you for that. I'm going to take that, if I may, as a comment rather than a question. Does that seem reasonable? Okay, that's a comment. I'm just making... Yes, please.
Me?
Yeah.
Thank you. Very thought-provoking comments, of course, but I think we need to see this quote and also the comments in indeed a historic context. These quotes were made when in Europe we came out dreaming of technology solving everything after the Second World War, and science at the time was being driven by white males, lone wolves, and to some extent also by the still existing colonialism. So if we want to solve the problems of the world today, and that would really now be the question how we could do that, and being inspired by Hans's talk and also by Manu's, I'm wondering whether we have to be more skeptical of focusing on technique and money and rather think about how we can use the resources that we have in the world, women, and democratization of the scientific process, because this will lead us to new ideas, new tools and techniques. I'm just thinking about using, for example, plants resistant to climate change that we can only find when we work with people who locally know about these plants. So my question should be, would be, do we not need to change our approach to science and make the whole procedure more female and more democratic? Thank you.
Also a comment, huh?
Well, we have 3 women on the bench here, as it were.
I take that also as a comment. And as you say, we have 3 females on it, and there were several sessions. It's great that we have so many women in science, and democratization is not easy, given the, at least in some fields, given the infrastructure that you need. It's also not that easy to do science, as most of us probably know. So, so the ERC, by the way, used to ask, as a criterion for judging projects, does it use a new technique? I ensured that we got rid of that question, because you can do fantastic science without using a new technique. If you have a bright and brilliant new idea that doesn't need a new technique, that's fantastic. Go and do it. And I think the most important thing you said was freedom to follow ideas, and that is what needs to be protected. But we come up against, I mean, when I started studying biology in Heidelberg, our botany teacher said, I don't understand all this need for all these fancy new microscopes. You know, I used to go out with a razor blade and a pen and a piece of paper and a pencil, and that was just fine for discovery. So no, it's not. You need new techniques because you come up against walls. We need both.
Thanks, Maria. Before I ask you, sir, maybe one question from Slido, which I think slightly addresses what we've just heard, which is new technologies can have a high barrier to entry, either in expertise or financially. How do we make emerging technologies more accessible to other scientists? And this is the democratization question that I was alluding to earlier on, where one solution might be to set up, for example, regional centers of technological knowledge and expertise. So there might be one in the north of England, there might be one in the west and the south and so on. Is that the sort of approach you might think of?
Yeah, I think you're hitting on a really good point that I think Nick also put into his opening comments, which is sort of bureaucracy as well. Yeah. That as science becomes more and more sophisticated, we're using more and more technologies that require huge funding efforts. That then puts the scientist in the purview of large funding organizations in order to do that work. And once you start building a large bureaucracy, that actually distances you from the public, distances you from the real world. And I think could be potentially what's underlying the problem now that we face with the skepticism, at least in the United States, of people to science, because it's run in these it's these white towers, these big institutes, et cetera. So what I think was very exhilarating to hear today was Manu Prakash's presentation, where his view is we need to, at the same time as we're pushing these new technologies, we need to assure people that what is being done in these big places is underscored by little things that people can do. The joy of discovery, the way that science is being done, sort of opening people's ideas to the reality that science can explain, has the ability to explain. So I support the idea that at some point, we're ready to go into big places with this new technology, but it's going to require lots of huge equipment that's not necessarily available to everybody, but with AI, potentially the data can be disseminated. But I think at the same time, we need to ensure that we have a robust mechanism for enabling, I don't want to say little people, but a broader swath of our population to be able to do science that is sort of reinforcing the fundamental concept that this is a strategy for understanding our world that has important consequences.
So I've got one question for you, Jennifer, if I may, very, very briefly, which is, you and I are both fortunate to work or worked in large, well-funded institutions with equipment and technology available. To what extent do you think such institutes should be permeable to others, and to what extent do you think they should not be?
Oh, I think it should be, there should be mechanisms, so it should be permeable. So one of the things that is very exciting for me at Janelia is, unlike at NIH, where it was very difficult to bring in visitors and outside colleagues, at Janelia, which is the HHMI campus, medical campus, there are mechanisms for bringing in people. They have a facility where you can come and use any of the microscopes at Janelia. And there's funding for that. Yeah. So you don't have to, pay for any of that. So I think we should try and encourage more of that type of effort. It requires the funding, the institute, the funding mechanism to be able to put money into that, but it's really, really important.
Yeah, I agree. Thank you.
Just quick addition, that exists here, of course, too. So there's the imaging center at EMBL, and there's the whole ESFRI's, European Science and Research Infrastructures. They're there, and that's great. It's not the same as having it at such an institute at your disposal all the time, as I can testify from my own time at EMBL. But I want to make one other point. I don't think we should obsess about this. And I talked about the more peripheral institutions that don't have access. I've always been wanting to write a piece, and I've not got around to it, called In Defense of Incremental Science. People use in sort of panels incremental as a derogatory term. It's not. It's absolutely needed. We need to make the steps. We need the confirmatory science. And we need steps that can be made with different techniques and that don't have to use these high-cost infrastructures. So, there's room for everyone. We're not a monolithic kind of culture. We have the variety. There's more questions out there.
We've very briefly, Nick, because we've only got 9 minutes left, and I want to make sure as many people as possible get a chance. But over to you. You sure? Quite sure? Okay. Sir, number 2.
Yeah, thanks very much actually for also having I think what Jennifer mentioned with the ivory towers and this public outreach is really relevant to my comment, which is in this sort of this rise of populism, whether across the Atlantic and in Europe. Can really scientists stay away from the nitty-gritty politics? I think Maria also mentioned how to justify to the taxpayers. How do then scientists, we can say in our circles, yes, we have these great ideas and we can let people do them for progress. How do we take that to the political sphere for where we need the funding, the sort of support to do it, and also, as perhaps on Nick's point, sort of keeping progress up in areas such as vaccine uptake, climate change, and all sorts of areas.
Yeah, thank you. Nick, would you like to respond to that, or Tamina?
No, I think that is really important. I'm not sure I have an immediate solution for how to do that. But I do think it's really important, following on from what Maria was saying about incremental science, also to think of the ways in which innovation doesn't just come from the big, well-resourced centres. Often, it can be the view from below, from the periphery, from the less powerful, and that might well include audiences that we're engaging who can provide views that have been ignored by those otherwise calling the shots and shaping the discourse. So I think science has a great deal to gain also from these processes of political engagement.
Thank you.
So actually, I'm really glad that I did spend some time in a lab that was very underfunded, where, you know, the most exciting essay that we did was a tripe blue exclusion essay. That was our go-to standard. But I can say that this sometimes makes you actually more creative. If you have less funding, less to work with, you just have to use your funding more wisely. You have to carefully think about the experiments you can do with that funding. And now, luckily, I'm in a really good position in the lab that I'm now in, and with my own PhD studentship. But still, I learned my lesson working there, and I'm glad for it, because still I think about doing things, considering carefully which experiments will be worth my while and using resources wisely. So I think actually sometimes it drives creativity and how to use resources carefully and well, if you don't have that much funding for everyone.
Thank you. I mean, I think I might comment too that my new activity and role at the Zoological Society of London says something similar, where on a shoestring compared with many other places, we are having real impact with relatively simple science, and it does make a difference.
Hi.
I beg your pardon. Hello, I didn't see you, sorry.
So people are talking about new technologies and all those things, but not much is done in the lab about the climate. If our labs are flooded, if they're on fire, what's the point of having these technologies for future? So I want the funding bodies to think about it and at least have like £50,000 to sort of give that lab for the sustainability. And I have a lot of plastic wash and reuse protocols and everything, and we have reduced our plastic waste to 90%, either washing and reusing or recycling, and I just want to spread that message. The only thing is we require funding for that, and same way with other initiatives. And I think people should start thinking about the environment in the lab as well, because that is very important for the climate change that's happening. And CO2, methane is also very important, the food waste that we see. So I think we have to now start thinking about the climate change and what lab can do. And I have an example in Dunn School at University of Oxford. So it would be good to sort of spread this message and people also start thinking about their science and environment friendly way.
So I think the COB certainly think about this, but I think it's incumbent on us all, not only in the lab, but everywhere we live and work, to think about climate change all the time. When I was at the Wellcome Trust, we've introduced a new health challenge, as we've called it, which is the effects of climate change on health, and I was pleased that we do that. But it's an interesting idea about support for labs in the face of challenges that are posed by climate change. And I don't know whether, Maria, that's something that EMBL or ERC would think about.
Well, EMBO has a prize for sustainable lab management. I don't know whether they'll repeat it next year. ERC is currently looking at what we can do. We're doing a survey of what other institutions, other funders are doing. It's not that easy, especially at the European level, because we come up against national programs, etc. But I thank you for your input, and feel free to write to me, and I'll bring it to the attention of the working group that is actually working on it and will make decisions within the next few months.
Thanks, Maria.
Thank you.
Over to Jeremy.
Hi, I've been really enjoying the discussion. So a quote that was mentioned earlier was also Sidney Brenner talking about how we're drowning in data and starving for understanding. And I can put this as in the form of really a question, which is, given that we do have so much data, actually, would it be fair to predict that, at least in the sort of tidal ebb and flow between ideas and technologies, that actually we've had a big period of incredible technology development, but actually in the next few years we're going to need a lot of ideas because what ideas do is organize the data. And is there perhaps ways of encouraging that, or talking about trying to make things more cognitively intelligible? Are there mechanisms or missions that we can set up to do that? It's an impossible question, but I thought I'd ask it anyway.
Anybody want to answer an impossible question?
Yeah, I mean, I completely agree with you. We've got to start trying to understand all of this data. The data is coming fast and furious, and we can very easily fall into a trap where we'll just say, well, AI, you figure out what's important. And what it will do is see patterns and relationships that aren't necessarily connected to the kinds of questions that we are familiar with. Now, that could be good or bad, but it could also come up with nonsense. And that's what I'm really scared about. So yeah, I'm definitely, this is something I think about a lot, and I don't have an answer for, but it's something we have to totally worry about.
Nick?
I mean, just to add very briefly, that could also be answered by thinking about science education and training and the skills that we encourage students to value in connecting the dots, seeing patterns, and developing concepts.
Yeah, but what are we teaching right now? We're teaching computational skills, looking, how to quickly move through your, different chat, systems.
Yeah, and there's still a great deal of
There's
memory work in
that,
practice,
yeah.
although it's derided by everyone. There's still a great deal of that.
Yeah.
Now, I'm deeply aware that Maria told me she has to go at 4:45 sur le dot. And so there's still some questions to come. So what I'm going to suggest, Maria, is that you discreetly take your leave. Bugger or whatever. So I hope you don't mind, but I know you need...
I'm really sorry to miss this. I screwed up with the time zones and got to get to the airport an hour earlier than I thought. So good fun, and hope to see you all around somewhere, and it was fun being in this discussion.
Thanks, Maria. Her own special round of applause. We can take, I think, 2 more questions. We have 2 people, and then we can sum up. But we're going to have to keep them short, I'm afraid, Madam. Off you go.
I'll keep this very short. I think it's, I lose a lot of sleep, primarily because, as scientists, we do have the capacity to see what's coming. We have a responsibility of showing to people what is coming. Everybody knows the sets of challenges. It's a very special privilege for us to do what we do, to pursue our curiosity. And frankly, I find we're not ambitious enough when it comes to the context of what science means to society and the mechanisms that can exist, that don't exist right now, in sharing the sets and tools and capacity. I mean, very simple example, open source hardware. If any sets of large organizations, funding bodies supported open source hardware, we would have a massive adoption of sets of tools and technologies at a pace much faster than it is currently. So in some sense, there's a lack of imagination. When challenges are quite large, we often forget to build completely new structures. And I mean, in some sense, we have exponential problems. We have to be thinking about exponential solutions. And those exponential solutions must include people. Otherwise, we cannot match. I mean, what's the point of carbon sequestration for, say, a city or a state, for example, of California? That wouldn't make any difference at all. So I think it's not as if paths don't exist. It's, open source hardware is one very clear example, but many other examples exist. I think we haven't put enough imagination in supporting much of the social side of this.
Thanks. I'm going to take that as a fantastic comment. And the final question of the meeting.
Thank you. Wow, that's a lot of pressure. I guess my question is to the panelists as well as to the company. In my opinion, the reason we are where we are is because no one understands what we do. I mean, you walk outside of this building, very few people would understand any of the presentations, any of the manuscripts that's been written in the past 10 years. So what are we doing to help the audience, the people who pay for our research, to understand what we do so that we can move forward?
Well, that's a really good question. And Tamina.
Sorry, is this my, yeah, it's on. So the master that I did in Dresden doesn't exist anymore. It has been replaced by a master in science communication. And the people who created that master program, they say that they've always been asked before COVID why they want to make this master's degree, why they want to create it. And since COVID hit, no one is asking that question anymore. Everyone's saying, oh, it's actually good that you're teaching students during the master how to communicate science. And I think the pandemic really has showed us how important it is that we can communicate it. I think there are lots of initiatives at universities around the UK to support science communication. Things like 3-minute thesis even make you think about how to communicate your science. And actually, in our institute, we've been talking about having something like a 3-minute thesis for the PIs, not just for the students. So I think everyone can do this as an exercise, but I just want to finish with a quote. You want to tell it? With a quote from Austin Smith that I got from him at the summer school I can really recommend. So Austin Smith said to us that if we can communicate, if we can tell our grandparents, our parents, our partner what science we're doing and why we're doing it, then that really means that we've understood the science that we're doing. And I think if we can all think about, if we're going home, how will we tell the people that we have in our life about what we've learned here, what we're working on, then this is already a good start, a good exercise that everyone can do.
That's a brilliant closing remark. I've just got a couple of things, if I may, to say. And one is actually that I think as important as being able to describe a piece of science is that people understand scientific method as well, that they know what an experiment is and how it should be interpreted. I think that's also important, and I don't think the public, whatever the public is, knows enough about that. But I'd just like to finish then by thanking the panel and Maria for their thoughts, comments and responses, you, the audience, for your questions and challenges, the Company of Biologists, as ever, for all their support. And I just want to end with 2 further quotes, which may be well worn, I don't know, but I heard about them from my PhD supervisor, Lewis Wolpert, like Sidney Brenner, a South African, and it was really about the futility of making predictions. So he said, as Paul Valéry said, like a man rowing a boat that floats on the tide, we enter the future facing the past. Our eyes only see the landscape of the past. What tomorrow looks like, nobody knows. And as Neil Bohr said, and maybe Yogi Berra, prediction is very difficult, especially about the future. So I shall stop there. I'm going over there.